All right, on to some exciting work happening in the labs at Dalhousie that could someday help determine the best treatments for some types of breast cancer. And my next guest is at the heart of this work. Dr. Paola Marignani is the principal researcher behind the work and the associate professor in the Faculty of Medicine at Dalhousie University. Good Monday morning, doctor. How are you?
Thank you. Good morning to you as well.
Yes, so very exciting news. Explain to us exactly how you came about this research.
Well, about 2005, 2006, we identified a new player in breast cancer, and this protein is called LKB1, and its primary goal is to suppress the growth of tumors. And so in essence, if you lose this tumor suppressor, you could potentially develop cancer. And so we began thinking about a model to develop, to test different theories or hypotheses that we had. And we generated a new model, a mouse model of breast cancer. And so in essence, what we've done is we've removed the gene, and the mice develop breast cancer quite rapidly.
Wow.
And so now we can actually, our discovery has now become a tool for us. So we can now test potentially new therapies in our mouse model. And hopefully the goal here is to try and suppress the growth of these tumors in the breast of the mice. And so it's really quite exciting for us, and I'm hopeful that it's exciting for everybody else as well.
Yeah, what was your first initial reaction when you found out that this happened?
Well, we weren't really, we had hoped, because it was a hypothesis at the time, so we weren't quite sure what would happen.
Yeah.
And when I went into the animal facilities to check on the mice, I started noticing tumors. And I was really quite excited. I tend to get quite animated when I'm excited. So I came back into the lab and I was a little stunned and a little quiet. And,
Yeah.
and then we started talking about it. And then we have to do all the experiments to follow up. And it was a very exciting moment for us.
And how long did the process take? How many years have you been working on this?
Well, the ideas started in around 2005 and 2006. Wow. Then we started generating the animal model in 2008,
Okay.
7 and 8, because there's a fair amount of protocol and procedure, and then we had to find resources, funding for it. And then by 2010, we knew we had something. And then it takes time to do, a thorough experimentation with all the appropriate controls and everything.
So I guess the big question is, now that it's in mice, when can you start testing it on humans?
Well, our goal now is, so we're into another phase now. We're beginning to test the various new therapeutics in the animals and in the cells that we take from the tumors. And once we know that we've got the right combination of new drugs, we hope to set up at some point a phase 1 clinical trial.
Right.
Right now we're in preclinical trials.
Okay. Okay. Very exciting stuff for sure. Hey?
I can't really tell you exactly when, hopefully within 2 years.
Yeah.
But again, with that, there's hurdles, resources, ethical protocols, and things like that, right?
I want to ask you if there's people out there, and I'm sure there is people out there watching who are suffering from or living with breast cancer or know someone who they lost to breast cancer. How should they feel at this news that this breakthrough has been made?
Well, I think there should always be hope. And with every new discovery lies the potential for new therapies and hopefully improved outcomes. I would just ask that everyone be cautiously optimistic, but optimistic nonetheless, and excited that it's a new day for research, and it's a new future for us, right?
Yeah.
I'm very hopeful.
Well, it's great that it was made here at Dalhousie too, right here in our own backyard. How has the community globally responded to the news?
Well, I think locally everyone's been really excited, and I've had significant support from local people and families, and from Canada-wide and globally. People are excited. I'm constantly fielding emails from scientists around the world asking if they could have samples or participate, or asking me a lot of questions that I'm trying to navigate through. So I think it's generated a fair amount of excitement for us, and I hope for everyone else as well.